Hello everyone, welcome to the AFOMP monthly webinar. Actually, as you can see, it's already the 6th webinar meeting from the AFOMP. As we know, year 2020 has come unexpectedly affected by the outbreak of the COVID-19, and the AFOMP has been offering during this hard time. AFOMP is thinking of offering online education for all the members in the AFOMP region. So we have honor today to invite Professor Aaron Shaw to give us a talk on the diagnostic reference levels. Today's moderator is Associate Professor Jenny Wang, who is from the Malaysia. I will give a brief introduction of Jenny Wang, and then he will he'll take over the webinar. Dr. Jenny Wang was received her PhD from the University of Wollongong, Australia. She started her career at the Department of Biomedical Imaging Faculty of the Medicine, the University of Malaysia, as a senior lecturer in 2011. She coordinated the UN Master of Medical Physics program from 2013 to 2017, which is accredited by the Institute of Physics and Engineering in Medicine, UK. It's the only postgraduate medical physics program outside of the UK and Ireland that has been accredited. The main research of Dr. Wang was included radiation dosimetry, radiotherapy, medical imaging, and radiomics. Dr. Wang has vast experience in teaching of physics and biostatics at the postgraduate level. She has been involved as a lecturer and examiner for the Master of Radiology and the Master of Clinical Oncology at the University of Malaysia since 2012. So now let's welcome Dr. Jenny Wang to moderate today's webinar. Dr. Wang.
Thank you. Thank you, Dr. Jim. I shall share my screen. Can you see my screen? So welcome everyone to the webinar, AFOMP webinar. So today we will be looking at the topic Diagnostic Reference Level, DRLs. It is a level used in medical imaging to indicate whether, in routine condition, the dose to the patient for an imaging examination or procedure, it is unusually high or unusually low for that procedure. It is a very practical tool to promote optimization of medical imaging examination. DRLs has been successfully implemented in relation to conventional radiography in the 1980s and subsequently developed for other modalities in the 90s. Well, many have heard of DRLs, I'm sure, out there, especially the clinical physicists. But perhaps not everyone knows how DRLs are established, the methods, what is the appropriate dose descriptor to use, and finally, how is it used as a crucial step towards optimizing the use of radiation in medical imaging. So today we are very privileged to have Dr. Arun Chogule to speak to us. Dr. Arun Chogule on this topic is the senior professor at and head of Department of Radiological Physics, SMS Medical College and Hospitals, Jaipur. He's also the former Pro-Vice Chancellor, Rajasthan University of Health Sciences, and Dean Faculty of Paramedical Science, Jaipur, India. He has 36 years of professional and teaching experience in medical physics. He's one of the pioneers in radiation biology and experiment dosimetry in India. He is the past president of Association of Medical Physicists of India and currently the president of AFOMP, as well as the chair of Education and Training Committee of IOMP. He has served as an expert to IAEA and has been regular associate to ICTP for 8 years. He has been awarded with numerous fellowships and awards, namely IOMP, IDMP 2016 Award for Contribution in Medical Physics, AFOMP Member Excellent Presentation Award, Outstanding Faculty Award 2019 of SMS Medical College, Dr. Farooq Abdullah Sher, E. Kashmir Best Researcher Award 2011 to 2012. Recently, he was awarded as one of the outstanding medical physicists 2020 by the AFOMP. So, Dr. Arun, we would like to listen to your talk on diagnostic reference level.
Thank you, Jenny Wong, for your kind introduction about me. Welcome to the participant for this monthly webinar. Can you just please unshare, Jenny, so that I can share? Okay, thank you very much. Just I'm sharing. So welcome to the AFOMP monthly webinar. And as has been talked by Professor Jin, because of the COVID-19, in-person meetings are not possible, and therefore we have switched on to the e-learning and online virtual meeting. Today's topic is Diagnostic Reference Level, DRL, Introduction, Concept and Current Scenario I will be talking about. Before that, please accept my greetings on behalf of me and AFOMP for forthcoming IDMP, which is celebrated on 7th November every year since 2013. And this year's theme is Medical Physicist as a Health Professional. Also, accept my greetings on the occasion of 125th anniversary of the X-ray discovery, 8th November 1895, X-rays were discovered. We are celebrating 125th year anniversary. Now, as you very well know, that since the discovery of X-rays were put into application for diagnosis and therapy. And today, every hospital, small or large, has a X-ray department or X-ray machine to start with. So radiation exposure in diagnostic radiology has contributed or is the largest contributor of the human-made, man-made radiation doses, and this is published by Unscare Report 2008, published in 2010. And according to some estimate, the exposure of patient to ionizing radiation from diagnostic procedure alone contributes over 90% of the total dose due to man-made radiation. About 30 to 40% of the US population undergoes one or more imaging tests per year involving diagnostic radiation. So the radiological procedure is being used very frequently for diagnosis. Even in India, about 80% of the population dose caused by man-made radiation is coming out from the medical diagnostic X-rays. Therefore, there is a growing use of diagnostic X-ray in the healthcare. And this is because of due to advancing imaging technology, we have started from a conventional X-ray to fluoroscopy to CT scan, then PET-CT, fluoroscopy, and so many the development has taken place, and more so in last 3-4 decades. A continuous growth in use of diagnostic X-rays has been recorded in recent time across the world in developing and developed countries. According to Unscare 2010 report, the total number of diagnostic medical examination increased from 2.4 billion to more than 3.6 billion in a period of 1997 to 2007. Means in 10 years period, there was a 50% rise from 2.4 billion to 3.6 billion. And the 2020 report is about to come, and it is expected that at least if you take 25% rise, there will be more than 4.5 billion radiological procedure for a population of about 7.5 billion. Means every second person is undergoing a radiological procedure. In a survey conducted in India in 2010, the number of diagnostic X-ray unit was recorded to more than 50,000, and every year almost about 1,000 to 1,500 new units are getting added. So global increase in the use of the radiation in healthcare, if you look at 3.6 billion procedure in 2008 as per the UNSCARE report. And even if you think of 25% increase in diagnostic radiology from 2008 to 2020, the number will be around 4.5 billion procedure. Similarly, the ionizing radiation is being used into nuclear medicine and radiotherapy. I will be speaking more about the diagnostic radiology and the diagnostic reference levels in diagnostic. Again, if you see at the UNSCARE report from 1988, 1993, 2000, 2008. If you just compare 2000 and 2008 report, the number of examination per 1,000 population has risen from 330 to 488, almost 1.5 times more in just 8 years. The procedure has increased. Similarly, if you look at the growth of number of diagnostic procedure and collective dose for 1,000 man-sievert, you will see that there's a 130% rise in the diagnostic procedures per this thing, and collective dose is 122%, whereas the population growth from 1988 to 2008 into 20 years is 26%. So much more faster rate than the population growth, the diagnostic procedures are increasing. Even again, if you look at per capita effective dose in millisievert from 1998 to 2008, this 2008, just 8 years, from 0.4 millisievert to 0.62 millisievert, means again 150%, that is 1.5 times increase in the dose. Also, UNSCARE report shows that the contribution of radiation dose to the population is 0.64 millisievert, whereas the natural background radiation contribution is 2.4 millisievert, means almost 25% of the natural background radiation contribution is from the medical exposure. Even there is another one study, NCRP report 2009 shows that in 1987 to 2006, almost 20 years period, the increase in medical diagnostic procedure or radiation dose level, the increase was a 5-fold in 20 years, that is 0.6 to 3 millisievert, highest dose increase in USA because of very high growth of diagnostic procedure. And therefore, as you know very well, that radiation is a double-edged sword. Radiation can cause hazardous effects, and therefore we have to keep the radiation dose as low as reasonably achievable. And therefore various approaches were thought, advocated and practiced since 70s, and there was a need was felt for standardization of a dose and reduction in variation in the dose without compromising the clinical purpose of each examination or procedural fail. That means one chest X-ray is being done in a part of a country, for example, from north to south or from east to west, the dose should not vary very much. And if it is varying, what are the causes, things we should look at. Also, there should be reduction procedure to bring it down by using various image receptor system or the image processing systems and technology development. So national survey in UK in mid 1980s showed wide variation in practices between the hospital itself, in one room to another room, the chest x-ray of a person giving variation in the dose of the order of 1 or 2 factors. And therefore, in UK, national reference dose first suggested way back in 1989. And now the concept comes, why do we need diagnostic reference levels in medical imaging? Now, you know, the general principle of radiation protections are justification. Any radiological procedure has to be justified that net benefit to the patient than the harm it is going to cause. And then if once you're justified that thing, you have to do optimization so that radiation dose to the patient as well as the medical staff is as low as reasonably achievable and dose limit. So optimization is a very, very important concept in dose reduction, which has led to concept of diagnostic reference level. So optimization, main concept of radiation protection, knowing and monitoring patient dose is a key importance for the optimization. And ICRP, International Commission on Radiation Protection, concept of optimization has led to put forth the idea of diagnostic reference level DRL, practical tools for promoting and implementing optimization. The requirement for establishing using DRL in diagnostic and interventional radiology has been introduced by IAEA into International Basic Safety Standard BSS, and IAEA for member states encourages to implement patient dosimetry, set DRLs, and use them optimization. So concept was brought by ICRP, taken forward by IAEA to the member countries. So the diagnostic reference levels were introduced by the ICRP way back in 1996 to help avoid patient dose that does not contribute to clinical purpose of a medical imaging task. So we have to keep in mind that the image quality is a diagnostic image quality information you have got. The dose should be as low as possible. And to identify those situations where it may be possible to reduce dose without compromising the required level of image quality, because our ultimate aim of radiological imaging is to extract the information from the image and arrive at a diagnosis, and therefore that factor is also very important. Keeping that in mind, you can bring down radiation dosage to be used as a benchmark for comparing patient doses resulted due to clinical practices, and thus they act as a trigger levels to initiate quality improvement. Means that if DRLs are set in a country, in my hospital, for a same kind of investigation, same range of a patient, if my doses are very high or very low, something is wrong. I have to look into that thing. That is a trigger level. So DRLs are not static and need to be updated on a regular basis with optimization or with change in technology. As you know, DRL values are available in the literature, but as the technology is changing, image receptors are changing, the image processing methodologies and the software is changing, the DRL will not remain the same. They will change as per the technology and updatation. Therefore, this DRL should be again evaluated every 3 to 5 years or whenever a technology change happens. As just I talked about that the concept of DRL was published in ICRP report number 60 in 1990, but elaborated publication from ICRP has come in 1996 about DRL, and further publication 105 has come up about that, and latest ICRP publication, which is devoted to diagnostic reference level in medical images, that report 135 has come in 2017, which I will be referring to for comparing, and how to this report gives you all the details, what quantity you should use, how to set a DRL, national DRL, regional DRL, local DRL, or intra-country DRLs. So subsequent recommendations of these ICRP were followed up by the International Atomic Energy Agency also, and IAEA used the term as a guidance level for medical exposure, but ultimately they have followed the same, that is the diagnostic reference level, changing from guidance level for medical exposure. And European Commission in 1999, EC has brought out DRL should be established both for diagnostic radiology and nuclear medicine, and if they are consistently exceeded, investigation appropriate corrective action should be taken. This has come into a European Commission law, and this is actually has to be followed by all the European member countries, and they are trying to follow implementing the things. National Council for Radiation Protection and Measurement in 2000 in United States, NCRP report 172, which defined diagnostic reference level and achievable doses for medical and dental imaging. Therefore, starting from ICRP, IAEA, European Commission, and then NCRP, all are endorsing and following the concept of DRL, and this is what international BSS 1996 and 2013, which has come, shows that those guidance levels, and then followed, I told you that DRLs should be implemented, should be used. So what are these DRLs are, or diagnostic reference levels are? Those levels in medical radio diagnostic practices. They are not applied to therapeutic procedures, because therapy procedures, the patient is getting benefited by the dose treatment and thing. In the case of radiopharmaceutical and nuclear medicine, levels of activity in case of the radiopharmaceutical and nuclear medicine for typical examination, for example, lumbar spine, fluoroscopy, or CT for a group of standard size patient, means adult 60 to 80 kg or different ways. If they are a pediatric patient, age groups up to 1 year, 1 to 5 years, 5 to 10 years, 10 to 15, or weight. So these levels are indicative of a good practice when not exceeded. For standard procedure, when good and normal practice regarding diagnostic and technical performance is applied, that is how we define DRL, diagnostic reference level. Now, how to set, how to proceed this diagnostic reference level? So what you have to do, patient dose survey and optimization you have to do, and you have to collect the data. I will talk about the detail, what quantities to be used, how to do it, just after a few slides that will be there. So collect the data on diagnostic reference level quantities for patients of 60 to 80 kg. Calculate the median dose from the examination, and then compare the median dose with if there are existing DRLs with that thing. If they are exceeding your value, median dose for the examination is exceeding, comparing with the DRL value set in for the country, then you have to review your diagnostic technique, exposure setting, and the equipment performance results for why are you getting the doses too high than the DRL. And then recommend optimization strategy and work with radiographers to implement the changes. And this example of audit is given into the ICRP report number 1 0.35, which is given, has come in 2017. So effective diagnostic reference level, which many countries are doing, and the UK survey report, what does it say? The DRL process has been popularized in Europe, and during successive surveys was shown to reduce the overall dose and the range of doses observed in clinical practice. So definitely implementation of DRL has helped to bring down, and I will show with some example, bring down the patient doses, staff doses. The use of DRLs in the United Kingdom and appropriate optimization resulted in significant reduction in patient doses, which has been reported by Heart D in 2004. In the United Kingdom survey of dose indexes, DRLs have been redefined every 5 years, and as I told, 3 to 5 years, you have to redefine and re-estimate and put it into the things. These surveys demonstrated a 30% decrease in typical radiographic doses from 1984 to 1999 in 10 years, and average drop about 50% between 1985 to 2000. Therefore, DRLs determined in 2005 were found to be 16% lower than those of the determined in 2000. So DRL estimation and the study definitely helps to bring down the radiation exposure and overall doses to the patient, to the public, to the population. So DRL major levels, as just I was talking about, the specification of examination of specific DRL facilities, comparison of practice, and so promote improvement towards as low as a reasonably achievable principle. And they can be national DRL set for large scale surveys as a tool to help identify usually high typical doses. And local DRL levels, they can be institute based, a city based, or a regional, the state based, set by each X-ray department as a characterize their typical dose level to facilitate the comparison with the national or the published data. So national surveys and registries for setting DRL values should normally include medium and large side healthcare facilities having sufficient workload. So when you are going to set the DRL values and the study you are going to do, the hospital you are going to choose should be a medium work base having sufficient workload, at least 30-40 X-rays of the different kinds. Say for example, chest X-ray, the other spine X-rays, lumbar spine, all kind of X-rays, at least 30 X-rays they are doing. And for large countries, a survey of random selection of small proportion of all healthcare facilities, a good starting point for setting national DRL values, results from 20 to 30 facilities are likely to be sufficient in the first instance. Smaller country with fewer than 50 healthcare facilities, initial survey of 30 to 50 of the institution will suffice to get a data. So you are going to collect data of almost 30 to 50% of the healthcare facilities in the country, and then finding out the median doses for each investigation, each kind of patients, it's adult, pediatric patient, if a pediatric age groups, or according to the weight. A survey of a particular examination in a facility should normally involve collection of data on DRL quantities for at least 20 patients. Means, for example, if I am going to find out chest PA view DRL, means 20 chest PA view patient of same adult, means adult weight 60 to 80 kg, those data you should get it, find the median, and preferably 30 patients, so statistical uncertainty will be reduced. For fluoroscopy and CT examination, and 50 patients for a mammography you have to take. So DRL values depend on the state of practice and available technology, including post-processing software. And as you know, we have moved from a conventional film-based radiology to filmless digital radiology, and where post-processing softwares are there, which affects definitely the requirement of the dose to get the required image quality. Median values, not the mean values, of the distribution of the data collected from a representative sample of patients within an agreed weight range that I told you, that 40 to 50, 50 to 60, 60 to 70 weight adult should be used for comparison with the DRL values. National DRL values should be set as a 75th percentile or third Q3, that is a quarter of the observed values. Regional multinational DRL values are created. They should be set as a median value of the national DRL values, and this has been done for the European Union because many countries in Europe they have found their own DRL for every country. They can be used for a regional, and they have been used for a regional multinational DRL values. Amount of radiation used for examination of a children can vary tremendously due to the great variation in patient size and weight from neonate to adult size adolescents. From few kg, 3 to 5 kg to up to 70-80 kg. There is a variation. Also, the size variation takes place. So the area required to be exposed for chest X-ray of a neonate of 6 months and 16 year will be definitely different, and therefore you have to make those groups and then find out DRL values. Weight bands are recommended for establishing pediatric DRL values. Age bands can be used if age is the only available measure. National and regional DRL values should be revised at regular intervals, 3 to 5 years that I have mentioned, or more frequently when substantial change in technology. You have moved from example from a conventional radiology to the digital radiology, or a image processing software high-end processor, or even now the artificial intelligence is coming in the diagnosis procedure. So that needs that the DRL should be again revised. If a local or national DRL values for any procedure is consistently exceeded, an investigation should be performed without undue delay. Is there any lacuna into the QA/QC, that is the exposure factors, the image receptor, image processing, and then appropriate corrective action should be taken, and that is, otherwise just finding a DRL or the exposure is not going to help. If they are exceeding, corrective measures has to be taken so that the mistake or the thing doesn't happen again and again. Now, just I told you that what DRL quantities you should use, what prerequisite you need to have when you are going to set the DRL locally, nationally, or regionally. So radiation matrix used for DRL should be appropriate to the imaging modality being evaluated. So when you are going to do the survey, same kind of machine, same kind of image receptor, same kind of processing. Otherwise, you have few X-ray machine which are low frequency, some are high frequency, somewhere it's an image receptor, image film based image, somewhere it is a digital imaging. That will give you very wide variation and the result will be wrong, and therefore appropriate imaging modality being evaluated has to be taken into, able to assess the amount of radiation applied to perform a medical imaging task. A particular medical imaging task should be easily measured or determined by the dosimeters. Establishment of DRL is generally based on surveys of DRL quantities for procedures performed on an appropriate sample of patients, and that I told you 20 to 30 patients, or mammography 50 patients. DRLs are typically set at 75% of the dose. distribution from a survey conducted across the broad user base, large, small facilities, public, private hospital, outpatient, indoor, using specific dose measurement protocol and phantom. So DRL should be set. The practical dose quantities used to monitor practice. The following are commonly used terms. For radiography, just simple radiography, you are going to do air kerma, air product pKa, or entrance surface air kerma. They can be used. For fluoroscopy and interventional radiology procedure, you need to use kerma area product. At the same time, you have to take into account the fluoroscopy time, number of images that has to be included into the observations what you are going to do. For CT, volume computed tomography dose index CTDI and dose length product DLP. For mammography, incident air kerma or entrance air kerma or mean glandular dose DG. For dental, intraoral radiography, incident air kerma or dental pKa for the dental panographic radiography. I will not go into the nuclear medicine. So these are the quantities suitable for setting DRL. For radiography, you can measure the doses in milligray or with a dap meter, milligray centimeter square. That is the dose area product. For the CT interventional procedure, CTDI or DLP, and for nuclear medicine, almost that is a amount of quantity of radioisotope in becquerel, megabecquerel, or becquerel per kg weight that to be used, and this is as per the ICRP report 137. So tool to identify those hospital using usually high doses based on third quartile values, mean hospital doses, ESD, DAP with DAP meter or CTDI, seen in national surveys and so already met by 70% of the hospital. Relate to typical practice, not individual patient. They are the broad, not for an individual patient. So limitation of published DRL. Now you will say, why should I measure or should I have a local DRL or my national regional? There are published values available in the literature. I can pick up and I can get those things. The published DRL values for other countries with potentially different imaging practices. What you are using, DRLs which have been found in another country, what kind of image receptor, machine, the image quality they use may not be relevant to your particular circumstances. The type of examination or a procedure specified for the published DRL may not be directly relevant to your particular practice. Published dose values may not have been obtained using the same methodology what you are practicing here, or same standard condition like the SETI dosimetry. Published DRL values may not be expressed in a different dose quantities or dose unit. The patient sample in a published survey may be different, and therefore you need to have your own DRL. Advantages in technologies such as post-processing and iterative reconstruction in CT will need to be taken into account when updating DRL, and that is what I said. If there is a technological advancement, you should update your DRL. So establishment of the DRL national and regional. DRLs may be established regionally, nationally, or locally from a data obtained from the surveys. Values of DRL quantities from patient examinations are collected from several different health facilities, and that I told you, level 1, level 2, level 3, private, university hospital, government hospital, having wide distribution from the country or the region. When national DRL exists for many countries with a region, European Union, just I talked about, the regional DRL may be determined by using the median value of the available national DRL, and this is what we use the third quartile or 75 percile for the DRL values. And typical values for the area and facilities surveys, if it's a healthcare facility consisting of several X-ray rooms or a small number of facilities or a single facility linked to the new technique, then median value of the distribution, as there are insufficient data to use the third quartile, a local use of to identify X-ray units requiring further optimization. So local, national and regional DRL can be set, and these are some of the requirements, and these are some of the application which they have narrated. So local implementation of the DRL, the dynamic, it is given by IPEM also. For each X-ray unit, establish optimized imaging protocol for each examination and patient group. As I mentioned, chest X-ray, what age group you have taken, okay. Measure typical doses as means for patient sample, compare with national DRL, compare set revised local DRL. If they are too high, periodic review 3 years or when the technology change. Now, how to measure this entrance surface air kerma, and here you can see it through this thing using TLDs or the solid state detectors, semiconductor detector you can use, and then you can measure the entrance surface dose ESD, or you can use a DAP meter here, and with this DAP meter what you will get is gray meter square or milligray centimeter square. So this air kerma area product gives you, because even if you change this distance, the because the radiation dose inversely proportional to the distance. If I double the distance, that it will become 1/4. At the same time, area will become 4 times, and therefore the product will remain the same independent of the distance of the dosimeter from the source of the X-ray. So DAP meter, and in European Union, DAP meters are mandatory for the X-ray unit, not yet in the Asian countries. When you talk about the CTDI or DLP, computed tomography, you have a phantom, the CT phantom, and you have the ionization chamber, minimum 10 centimeter long, the ionization chamber, and then you measure for a single rotation or multiple rotation with the product. So this will be DLP, dose length product, and this will be just the CTDI for volume. For a mammography, mean glandular dose with the dosimeter you can do. So ICRU DRL quantities for CTDI, volume, computed tomography dose index volume, DLP for dose length product, for this incident air kerma, mean glandular dose for the breast, and so many quantities you entail with a different kind of investigation. So recommendation for DRL: the use of diagnostic reference level as an important dose optimization tool is endorsed by many professional and regulatory organizations, including ICRP, American College of Radiology, AAPM, United Kingdom, Health Protection Agency, IAEA, European Union, Australian organization. So there has been an erroneous tendency to assume that being below DRL means adequate optimization, and this is wrong. Many people see my doses are less than the DRL values, but you have to see that DRL values for which condition, for which kind of procedure, what set of patients they were used. So just DRL is a one step in the optimization. What are the limitations of DRL? Most dose surveys for DRL have assumed acceptable image quality, but they have not defined what is image quality on a objective base. It's just subjective that yes, acceptable image quality. So you have to take into account objectively the image quality. How many DRL related papers have mentioned that we selected X number of images detected? So that is not a correct way to do, that's a limitation. So going beyond DRL you have to do, so you have to define the image quality. and that image quality objectively, not a subjectively. What if we start with image quality and make it priority? Only if image quality is acceptable, we move further with a dose, mean, median dose, or any level for the dose. And you have to go beyond standard size. Each healthcare facility should establish specific protocol for each patient group as part of optimized practice. Protocol for pediatric examination, patients grouped by range of weight or age, taking into account changing technology. Fast developing technologies may drastically change the patient dose levels. Either they may increase or decrease, and therefore they will impact the DRL. So benefits of the DRL are image quality primary if you take it, dose secondary if you take, can overall weight group can be applicable to optimization of the individual. And here you can see for a pediatric patients, the weight groups less than 5, 5 to 15, 15 to 30, or age groups they have been taken 1 year, 5 year, and this is what recommended weight groups for the body examination, less than 5, 5 to 15, 15 to 30, or age group less than 3 months, 3 to 1 year, 1 to 6, more than 6. So this is what has been published and has been in the literature. So how to establish DRL? Establish a working group, decide for which imaging procedure you want to set a DRL, decide dose quantities, decide methodology, patient group, sample size, and are you going to do on a patient or on a phantom, prepare data collection form, can be electronic or paper-based, collect data from contributing X-ray rooms, analyze the data, find the medium of the DRL for large hospital data from different X-ray room for some procedure to establish local DRL, statistical analysis, set DRL usually third quartile or 75th percentile. So points to remember, DRLs are typical dose values for standard size patient for a particular examination. Keep in mind that they are for a standard size patient for a particular examination. DRLs are representative for a practice in a country, and that is what I was mentioning, published data from another country. For example, if you look at the DRL for the chest PA view in Europe, UK, US, and Japan, you will find that they range from 0.3, 0.2, 0.15, almost double range, and therefore you should have your own DRL for a country specific, region specific. DRLs are indication of amount of radiation used for specific procedure. DRLs are a tool for optimization of the protection. DRL values are not intended for individual patient. They give just the range of the doses. DRLs are not dose limits. DRLs are not border between good and bad practice. It varies. DRLs are not static. They should be revised periodically or as and when the technology changes. Dose audit should be performed in each medical facility and local values compared with DRLs. Local review if dose above or substantially below DRL. Optimize of clinical protocol. DRL should be included in the education and training programs of the health professional involved in medical imaging. Patient dose audit and DRLs are important tool for optimization of the patient protection. I need to stop here, but because I hope my time is over, but these are the published values. They are available in literature. They can be used for various radiological procedure from different countries, for a pediatric, for different age group from different countries like Europe, UK, Germany, Switzerland. They are available. In conclusion, DRLs have proven to be a useful and valuable tool for optimization of radiological protection in medical exposure of patient. All individuals who have a role in subjecting a patient to a medical exposure should be familiar with the DRL process as a tool for optimization of the protection. Application of DRL is not sufficient for optimization of the protection. The diagnostic quantity of corresponding image must also be evaluated. DRL value should not be used as a dose limit. Dose limit do not apply to the medical exposure of the patient. National and regional DRL should be revised every 3 to 5 years or whenever the technology gets changed. It is required that both researcher and government agencies come forward to work for setting up the DRL. Not having the national DRL, there are many reasons to do that. Lack of appropriate infrastructure, medical physicists not sufficient, so many things are there. I will not go into the details of this thing. I will stop it here and I will like to take questions from you. So once again, thank you very much for patience hearing and kind introduction. Thank you very much. I stop sharing. Thank you very much.
Dr. Wang. Thank you, Professor Allen. Dr. Wang, yeah.
Thank you. Thank you, Dr. Arun. Any question from the floor?
No, you have to take question from the chart, I think.
Yeah. So there is.
Okay. So there is one question. How to define
how to define unacceptable image quality to define DRL? And can you recommend any objective measure for each diagnostic product?
Yeah, so that is what I was talking about, that image acceptable just is subjectively is not sufficient. So on a scale of 0 to 10, how the image quality, the optical density, the image details, the resolution, based on that thing, you have to score your image, and what dose is required to score that on a point 0 to 10, that is required for different kind of imaging systems and different kind of imaging to arrive at. That is a subjective definition of a image quality and not as a vague just saying image is acceptable, because one radiologist the image may be acceptable, for another radiologist that may be a poor quality or may not be not acceptable. So that becomes a subjective. So objective criteria based on the image quality has to be done on a scale. Any other question?
Yeah. There is one more question. How relevant it is to adopt the DRL established by others in their settings or hospital settings?
No, what is the question? DRL setting? Just let me check.
How relevant it is to adopt the DRL established by others in person's own hospital or clinical settings?
That is
what should be the criteria?
That is what I mentioned, is that if you are going to follow or you are going to take the DRL which has been set by a different country or different institution, then you have to make sure that the DRL for which set of examination, what kind of the image processing, image receptor and the X-ray machine, if it's a similar, you can follow. Or also, if you see a country, various countries, the patient size, the required exposure factor will be different, and therefore while taking you have to be cautious that the image parameter, the techniques what you are going to use and the patient parameter are nearly similar, you can. Otherwise, you have to have your own institutional or country, because country to country, the patient size and also the technology may vary, and therefore you have to be cautiously follow.
Yeah. Thank you, sir. We have one more question from Malaysia. It is, what is the standard number of sample size for any radiographic procedures for the DRLs?
Yeah, that is what I told, that minimum 20 to 30 procedure. For example, I told you, if you are going to find out the DRL for the chest X-ray, then on that machine, similar kind of patient, means weight you have taken 60 to 80, or maybe 40 to 60 or 50 to 60, at least 20 patients, but preferably 30 are there, statistical uncertainty will be reduced. So you have to have that thing. Similarly, when you are going to have a national DRL, that means at least 30 to 50% of the X-ray machine or installation, and every X-ray machine 20 to 30 investigation of the same kind has to be done. So very, very large data you have to collect for a national DRL.
Okay. Thank you, sir. We have one more question. They are asking, when publishing facility DRLs, do we use median as the local DRL? And when we compare to other country DRLs, do we use the facility median or the 75th value?
It is recommended by the ICRP to use 75th percentile value when you are going to compare the nation to nation or some other country values you are going to do. It is recommended that 75th value should be used, percentile value should be used.
Yep. So now I hand over to Professor Jenny for
Thank you very much. Thank you very
further.
much.
Thank you, sir, for such an interactive talk. Professor Jenny, you can unmute yourself.
Yeah. So thank you, Rajni, for bringing the question. I have some question as well. If when we get in the CT audit for DRLs.
Okay. You're muted. I'm not able to listen to you.
Hello?
Yeah.
Yeah.
For,
Is this?
Yeah.
For CT?
When we get for CT, we try to get the CT dose screen to establish the DRLs, you know. But we find that
not able to listen.
Again, your voice is not
voice is breaking.
audible.
Study,
Yeah.
do you have any practical suggestion on how to approach DRL?
In between your voice was not there, so I couldn't understand your question. You said CT audit, but next your voice was not there.
Okay.
No, some voice problem.
Yeah. Professor Jenny, you can write your question in chat box, so we can see from there.
So
Yeah, you can type.
Yeah.
Okay.
Excuse me, can I ask a question, please?
Yeah, sure, please.
Yeah, sure.
Yeah. Prof. Arun, hi, how are you? Thank you for
fine.
the wonderful presentation that you have done with us. So I'm asking here, for example, if you are using the hybrid imaging techniques, for example, like PET-CT scan. In that case, are we going to establish a DRL for the nuclear medicine alone and the CT, or together?
Yeah, very good question. It has been recommended that if you are going to use a PET-CT, for CT separately DRL, for PET separately DRL has to be established, has to be followed, not a combined one, because for CT you have CTDI and or DLP. For the PET you have the isotope megabecquerel or megabecquerel per curie per kg, and therefore separately these two imaging modalities are different. That is, one is the X-ray based, so they have to be found out differently, reported separately. Very clear. Very clear.
Pronunciation or more different.
Any other question?
Yeah, I found that Dr. Jenny's, Dr. Wang's question. She said that we have experience with getting the date from DICOM those screens.
Screen, but those screen data.
Yes, it understand the standardized between vendors. The format is also not easily
exactly
passed.
Exactly. See, what they give it from the vendor dose is actually on a Monte Carlo, and the parameter what they use actually, isn't it? It is direct measurement, and therefore you have to do on a phantom measurements and then compare actually.
Okay.
Used by the different companies are different.
Okay, I think the times it's close. It's approach 4 o'clock. So thank you. Thank you very much for Dr. Allen for the wonderful talk, and I will give more information about the next talk, next webinar. So as we know, there's some information. So our next webinar will be on December 10th, 2020. It's also Thursday. Our speaker will be Professor Sharman. So his title will be Recent Advances in Break Therapy and the Challenges. So hope to see you next month. Also, we have actually scheduled our webinar from next year. So it's from January to 2021 to July 2021. That's also being scheduled. We will release this information for next month or month by month. So hope there's not much to adjustment. Another information is our webinar will be sponsored by the continuous professional development of the Asia Oceanic Federation Organization. So this actually was endorsed by the Australian College of Physical Scientists, Engineering and Medicine. So each time you listen to the webinar, you can get 2 points as long as you register through this link. I also have pasted this link on the web on the chat line. So, but hope you do not spread this because as only we actually have 2 ways to certify your participation in this webinar. We have in our back date we have your time, how many time you spend on this webinar. So this is just for someone may need the points, someone we don't. So if you need the certification points, you can just log in and put some of your information so we can certify you have listened to the webinar. So that's basically all the information we release. So and at the end, so let's thanks again for Dr. Allen for your talk. So hope to see you all next time.
Just Jin, one information that we have on the 7th November, we have AFOMP IDMP webinar on the 7th. Already the information is on the AFOMP website. So all the participants are requested to join to the AFOMP IDMP celebration on 7th November.
Okay. Regina, are you going to share this information?
Yes.
Yeah, good. Can you share?
I have already shared the screen, but I'm not.
That's not opening.
Let me see. Do I have one?
I have already shared the screen, but I don't know why it is not visible.
Okay, so you have to unshare, I think, again, and then
Yeah.
Bin can say it. But okay, already it is on the AFOMP website also. All details are there. Yeah, he is able.
Is it visible now?
Yes,
Yeah.
It's come up.
Yeah.
So this is our program, and this is the link. It is available on AFOMP website, and this is the program for the day. So we are hoping to see you all.
So same time, actually.
It's the same time what we use for AFOMP monthly webinars.
Okay. If there's no more information, we are going to close today's webinar. Okay. See you.
See you.
Thank you for joining and thank you for talking. Thank you. Bye-bye.
Thank you all.